So now what I want to talk about, what are known as the light-dependent reactions. This, of course, is the first stage of photosynthesis, and it's going to take place in the thylakoid discs. So if we look at the thylakoid membrane, okay, this is the stroma up here. This will be the thylakoid space. Okay, if we're looking at the entire process of the light-dependent reactions, we're going to see several things. The first thing we're going to see are two distinct structures that look like this. Okay, and these structures are jam-packed with chlorophyll pigments. And each has a specialized chlorophyll in its center. Chlorophyll pigments chlorophyll pigments okay, and these regions, or these protein-like regions containing the chlorophyll pigments, are called light harvesting complexes, and they go all the way around in this entire structure. And this whole structure, including those light harvesting complexes, a specialized chlorophyll pigment, and a reaction center complex, is called a photosystem. And we have two different photosystems. We have photosystem 2, which comes first, and we have photosystem 1. The specialized chlorophyll inside of photosystem 2 is called P680 when it's reduced. And the specialized one inside of photosystem 1 is called P700 when it's reduced. Now each of these specialized chlorophyll pigments obtains its electrons that reduce it from its non-reduced form from a different source. For P680, a water molecule will be split into molecular oxygen, protons, and of course 2 electrons. So those 2 electrons from the water molecule each will reduce P680 plus to P680. Okay? Before these electrons are passed from P680 to any other molecule, what happens is sunlight hits and excites electrons in the chlorophyll pigments in these light harvesting complexes, causing them to enter an excited state. In that excited state, these electrons have higher levels of energy, and as they fall back down to their ground state, they begin to release the energy in 2 forms, as heat and as light, which we call fluorescence. So the light energy is transferred from chlorophyll to chlorophyll until ultimately it's transferred from one of the final chlorophyll pigments in the light harvesting complex to our specialized chlorophyll pigment in the center of the photosystem, exciting the electron there. When 2 electrons have been excited from P680 and enter the primary reaction complex, the reaction center complex, and the primary electron acceptor in that reaction center complex, they're then passed down an electron transport chain and ultimately wind up reducing P700. So P700 obtains its electrons from the electron transport chain, whereas P680 obtains its electrons from water. Now, before we proceed into what's going on in P700, remember that as these electrons are passed down the electron transport chain, they result in the pumping of protons across the membrane. So protons get pumped from the stroma into the thylakoid space, generating a proton gradient. Just like what we saw in respiration. So we now have this proton gradient here. Okay. Next, before we get to that proton gradient, let's go ahead and follow these electrons. So again, light energy going to be transferred from pigment to pigment in the light harvesting complexes until it ultimately excites the electron in P700. That electron enters an excited state and is transferred to the reaction center complex. When 2 electrons have been transferred there to a specialized protein called ferredoxin, those 2 electrons are then ultimately transferred to a specialized enzyme called NADP+ reductase. That is involved in the reduction of NADP+ to NADPH. And you'll remember that NADPH is one of the molecules required for the Calvin cycle to proceed. So we've produced NADPH. Notice how we did that. To get the electrons to the excited state needed to ultimately reduce NADP+, we needed to accrue the energy from sunlight, both in the photosystem 1 and photosystem 2. And it was the combination of the light harvesting complexes through both photosystems that ultimately allowed the electrons to be energized enough to reduce this to this. Without both of these photosystems, we would not be able to do that. Okay, and the final part of this whole process involves a familiar player we've seen before, and that is the enzyme ATP synthase. Again, it looks like this. Okay. These protons move in, occupy space, causing, as they occupy, the molecule to spin. Eventually, protons are released from it as well. And down here, that causes the molecule again to spin. And as it does, it generates molecules of ATP from ADP and an inorganic phosphate. And this ATP is the second molecule required for the Calvin cycle. So both the NADPH and ATP are produced in the stroma, and they're going to be used ultimately in the Calvin cycle. Okay, so these are used in the Calvin cycle. Now, this that I've drawn on the board currently, using photosystem 2 and photosystem 1 along with the ETC, is an example of linear electron flow. The electrons start over here, and they flow in a linear fashion over to here. But there's a second kind of electron flow, and it's called cyclic electron flow. Cyclic electron flow only uses photosystem 1 and the electron transport chain. The electrons are not ultimately passed to ferredoxin at the end of this whole process here. They do not ultimately reduce NADP plus to NADPH. Instead, they're passed back into the ETC. The benefit of this is that we can endlessly carry out the electron transport chain, generating a proton gradient, thereby giving us ATP. So cyclic electron transport is a good way to make additional ATP that might be used in the Calvin cycle. However, it is not a good way to generate NADPH, as none is produced during cyclic electron flow. So, linear uses photosystem 2, the ETC, photosystem 1 produces NADPH and ATP. Cyclic just uses photosystem 1, the electron transport chain, or at least a part of it, and only generates additional ATP. So these are the light-dependent reactions.